Good evening, all.
Yes, sir.
Hello. Hello. Right. Discuss gem used in completed some endonucleases. Now another important topic: modifying enzymes.
Sir, voice is not audible, sir.
Now clear?
Yes, sir.
Hello. Right. So first, let's discuss about the ligages. Ada told you last class what are ligages? What are the function of ligase enzyme?
Pasting of the enzymes, of joining.
Joining. Wait. Joining of enzymes. Okay. tDNA ligase, T4 ligase. No, this one not. Right. Is it clear? Hello? Hello?
Sir.
Voice is clear first?
Yes, sir.
Right. So ligages are used covalently joined, already told you, joined two fragments by creating phosphodiester bond between 5' phosphate and 3' hydroxyl group of the adjacent nucleotides. Right? We can observe here clearly. So covalent bond formation, 3-hydroxy here, 5-phosphate. This is 5' phosphate group. 5' phosphate group, right? They mainly for that this DNA ligases require ATP energy. They require main ATP energy and magnesium ions. The most commonly used ligase is the bacteriophage T4 DNA ligase. There are different type of ligases are there. 2,4 DNA ligase and DNA ligase E. coli, TOC DNA ligase. They are and T4 RNA ligase. So these are different type of ligase. Among these 2,4 DNA ligase, the most important one. And other ligases are E. coli or DNA ligase and TOC DNA ligase and bacteriophage T4 RNA ligase. Right? The DNA ligase is primarily responsible for joining the gaps that form DNA during replication. What is replication? What is replication? Hello? What is DNA replication?
సేమ్ ఐడెంటికల్ కాపీని తయారు చేయడం.
Right, in DNA replication, this is, for example, this is two strands, red color two strand, right? One is 5 end, another one is 3 end. The opposite direction always 3 end and 5 end, right?
Yes, sir.
Right. Now in DNA replication, during the DNA synthesis, first of all, change color. So here first one always the 3 to 5 end is template. So here the 3 end. So 3 end new DNA strand synthesized from this 3 end ends towards the 5 end. And in above, this is A stand, this is B stand. Now B stand synthesis completed, right? The B stand mother stand direction 3 end to 5 end, okay? Hello?
Yes, sir. Yes, sir.
3 end to 5 end. What about the opposite direction stand? Opposite
Five
newly
prime.
synthesized stand.
5 and 2.
Right. This is 5 and 2, 3 end. Always 5 and 2, 3 end. Now here, the what about other side? That is the A stand, A DNA stand, 5 and 2, 3 end. Newly synthesized DNA from the end, this end towards the.
Okay.
So this is 5 end and 3 end, right? So here the newly synthesized stances, one is green stand and another one is blue stand, right?
Yes, sir.
Blue stand and blue and green stand. So here what happened? The always newly synthesized DNA, for example, this is the 3 end. Starting point here 5 end. So continuation 5 end to 3 end. This is continuation stand, right? But here the opposite direction. Where is 3 end? This end 3 end. But it does not shows the continuous stand. It shows the breakage. Breakage means synthesize some distance and break it and again synthesize some fragment. Like this number of fragments are formed. This is called DNA replication. Let me take the whiteboard. I will explain clearly. Right. 5 and 3 and okay. Opposite strand 3 and 5 and. Is it clear? First, this is DNA strand, mother DNA.
Yes, sir.
Newly synthesized DNA. This is the 5 end strand. It shows continuous towards the 3 end.
3 and.
But what about here? Please be silent. Hello. Please be silent. Thank you. Five and 3 and 3 and 5 and. For example, this is act as a template. 3 and this 3 and act as template. So first 3 and 5 and 2 direction goes only this direction only. Later this winding DNA unwind, separate it, and shows continuation up to the end. But what happened here? Opposite direction. In opposite direction, only the synthesizer stand from the one fold. This fold. This way. Five and 3 and. Opposite 5 and 3 and. For example, this is the first helix. First helix, second helix, 1, 2. Okay. This is 5 and, right? Where is 3 and this for this 5 and? Opposite direction. Hello?
Yes, sir. Yes, sir.
So this 5 and 2, 3 and new strand formed. Is it clear first? Hello?
Yes, sir.
First, this is DNA double helix. Does not form any DNA. Actually, these 2 copies synthesize another new 2 copy. But one is continuation strand, another one is discontinuation strand. Continuation called as leading, discontinuation strand called as logging. Continuation strand leading, discontinuation strand called as logging. Let's see how the formation of leading and logging strands. First, so here the 5 and direction. So newly synthesized from this fork only, from this direction. Because always the 5 and opposite 3 and, right? So the synthesize, newly synthesized always 5 and to 3 and. 5 and to 3 and. So where is 5 and in this fork? This is the first 5 and. This is clear, first one. 5 and opposite should be 3 and. Don't get confused. Right. So now this stand, 3 and stand. 3 and stand, newly synthesized stand, begin from the open or closed stand. This is the open area. This is the closed complex area. Now the newly synthesized stand begin from this direction because this is 3 and. So opposite it should be 5 and, right? Hello?
Yes, yes, sir.
Right. So one blue color down, green color up. Next, after separated one helix, it means this helix separated. This one, this region unwind. Unwind. Now this as it is blue color as it is, right? And next gap formation after unwind. Another blue also formed. So this is the 5 and 3, right? Another blue formed later. What about green color? Green color continuation first fork. And again it shows without break continuation to the next fork. Like that it is continuously extend toward the end of the DNA strand. So here the one fold, two fold, here the formation two breaks. One fold for one break and another fold for another break. That's why the newly synthesized DNA formed into breaks. So this breaks called as Okazaki fragment. So one is without break. Green color without break, this is called leading. And blue color shows the break, that is called logging.
Logging.
So this logging breakage, called as Okazaki fragment. Okazaki fragment. So the main functions of ligase, this gap. So between the Okazaki fragments, the gaps are formed, right? This gap filled by or attached by, joined by ligases enzyme, and also repair the DNA. So although the DNA and RNA ligases catalyze the formation of phosphodiester bond between adjacent nucleotides, and also not only fill up the gaps and also fill up the phosphodiester bond. Here are the two type of repair, gap repair and nick repair. Gap repair means, hello?
Yes.
Gap repair means here the from one or two nucleotides missed in DNA. That is called DNA gaps. This is called gap. This is also called gap. And this gap can be repaired with help of DNA ligases enzyme. And also DNA and RNA ligases catalyze the formation of phosphodiester bond between two nucleotides. And in nick direction, in nick here formation mainly, it is not a mixed of nucleotide present, adenine, thymine, guanine, they are all present. But here missed between the phosphodiester bond. Do you know DNA structure? These nitrogen bases attached with phosphodiester bonds. So this phosphodiester bonds attached by DNA ligases and RNA ligases. This is called nick repair. And gap repair added new that means the missing nitrogen bases added. So they are all done by DNA ligases. And let's see the first, the ligase catalyze the formation of phosphodiester bond between adjacent 5' and 3 hydroxyl terminal. And in a sense, the opposite restriction endonuclease, but they are not influenced by the local sequence. That means actually, what is the function of restriction endonuclease? What is the function of restriction endonucleases?
Cutting, sir. It helps to cut.
Cutting. But what is the function of ligases?
Filling the gap.
Filling the gap. So these both are opposite function. Here the ligases require either ATP or NADP plus as a cofactor. And also sometimes utilize the magnesium ions also. But what about the restriction endonucleases? Restriction endonucleases may be used cofactor or without cofactor also they can done their activity. Right? Here the different type of ligases are given. In this the first one T4 ligases. T4 DNA ligase isolated from T4 bacteriophage. It is commonly used in the constructions of recombinant DNA molecules for molecular biology application. What is rDNA? What is DNA molecule? What is DNA molecule? Hello? What is recombinant DNA?
The newly formed
Recombinant.
అంటే జీన్ ట్రాన్స్ఫర్ అవుతాయి కదా సార్.
ఎలా ట్రాన్స్ఫర్ అవుతాయి? How they are transferred?
అదేంటి?
Two vectors. Host.
Conjugation. Link. Cross. Crossing over lo.
Right, crossing over. In crossing over, two different genes are recombined. Right? Let's see T4 DNA ligase. T4 DNA ligase. No. Right. In T4 DNA ligases, here ligase enzyme, so ligase enzyme, so joining 3-hydroxy group with 5-end phosphate group. Covalently bond. Here this is 3-end and this is 5-end. Due to covalent bond formation between 3-end and 5-end, what happened finally? You can observe here, phosphate bond formed. And the DNA sealed by ligase enzyme. Now this is open DNA. That means here the nucleotides present, but phosphodiester bonds not formed. Now this here the phosphodiester bond attached to in the form of ATP, the ATP triphosphate. One of the phosphate given to the bond formation between hydroxyl group. As a result, this ATP convert into adenosine diphosphate and form phosphate bond. As a result, the DNA strand is sealed. Right? Hello?
Yes, sir.
Ligates the ends of duplex are DNA and RNA. So this activity done in either DNA and RNA. Join blunt ends terminal as well as and with cohesive complementary. Blunt ends. Already told you what are the blunt ends. What are the blunt ends? Blunt end means there is no cohesive ends. This is blunt end. These are cohesive ends. So these both ends are attached with ligase enzyme, and it requires both magnesium ions and ATP for activity as a cofactor. And already told you initially what are the requirement for ligase enzyme? Ligase enzyme activity, magnesium ions either ATP or NADP. In ATP, NADP, any one act as a cofactor. But in this T4 DNA ligase, it require both magnesium ion require and ATP require, but cannot use NAD. Right? E. coli DNA ligase, in E. coli DNA ligase, but in E. coli DNA ligase, which like most prokaryotic DNA ligase uses NAD as a cofactor instead of ATP. So T4 DNA ligase, which is extracted from the bacteriophage, uses ATP instead of NAD, but E. coli extracted DNA ligase using NAD instead of ATP, right? Is it clear? Hello?
Yes, sir.
Right. However, unlike T4 DNA ligase, the E. coli DNA ligase enzyme does not show blunt-ended ligation activity. Here the T4 shows the activity between blunt end and cohesive end. But here E. coli does not show the blunt end ligation activity. The DNA ligase has become the more widely used in DNA manipulation, right? The T4 DNA ligase and recently known TOC DNA ligase are used to join adjacent single-stranded DNA, right? So anyway, in T4 DNA ligases, it can use in DNA and RNA molecules binding, and the requirement mainly magnesium and ATP without NAD. This is the most important. And here the joining of hydroxy phosphate group utilize ATP. Already told you, in ATP one phosphate utilized and converted into ADP, right? The repair single-stranded nicks in duplexes, DNA, RNA, and DNA, RNA duplexes require ATP as a cofactor. Next one, DNA ligase E. coli. Almost similar. The particular of the enzymes here catalyzes phosphodiester bond between duplexes DNA with cohesive end. What about T4 DNA ligase? T4 DNA ligase also catalyzes phosphodiester bond between duplex DNA. Apart from the duplex DNA, duplex RNA also. Duplex DNA and duplex RNA also. But what about DNA ligase? Hello? Respond, anyone? Are you there?
Yes, sir. Yes, sir. Yes, sir.
I am comparing with T4 DNA ligase with E. coli DNA ligase. First, are you clear with T4 DNA ligase? T4 DNA ligase. What are the requirement for ligation activity of T4 DNA ligase?
ATP and
Right, ATP and magnesium. Write down one side.
ATP.
First one page, write down T4 DNA ligase. And next place, write down DNA ligase E. coli. And next phase, write down TOC DNA ligase. 3 enough. Make it compare. It will be easy to you. So first requirement T4 DNA, as you said, magnesium ATP. What are the requirement in DNA ligase?
ADP.
No, NAD plus. NAD plus instead of ATP, here utilize NAD plus as a cofactor. Right? Next, first T4 DNA ligase. In first one, catalyze a phosphodiester bond between duplex DNA, duplex RNA with blunt end and cohesive ends. Blunt end and cohesive end. But in DNA ligase, this same catalyzes phosphodiester bond formation between only duplex DNA and with cohesive ends only, not blunt end. It will not efficiently ligate blunt-ended fragment. And it requires only NAD plus as a cofactor. Same mechanism, same. Mechanism same means, what is the mechanism in ligation? In ligase reaction?
Filling the gap.
How it is gap filled?
Phosphodiester between 5' and 3.
Between 5' and 3'. Here the 5' phosphate, 5' phosphate joined with 5' phosphate joined with where?
3'.
3' what?
Hydroxyl.
Which group?
Hydroxyl.
3' hydroxyl group. Here the which bond formed?
Phosphodiester.
Covalent bond formed. That means of course phosphate, but it is bond formation between OH and phosphate, covalent bond, because here the sharing of electron. That's why it is covalent bond formation between OH and 5' phosphate. But in DNA mechanism same, does not change the mechanism in DNA ligase E. coli or T4 DNA ligases. Mechanism never change. But change is in the T4 DNA, duplex DNA, duplex RNA, or single stranded RNA, single stranded DNA, these are all repaired, that they are attached and with blunt end and cohesive end. Whereas in DNA ligase, only duplex DNA with cohesive end only. And it utilized only NAD plus as a cofactor. Next, TOC DNA ligase. TOC DNA ligase used to join adjacent single-stranded DNA, not duplex. Single-stranded DNA molecules that have been polymerized and a template from primers. That means, actually, this is the template. This is newly synthesized strand. Whenever, if any nucleotide missed or any phosphate bond or phosphodiester bond fill up is missed, in this situation, the TOC DNA ligase utilized. That means the TOC DNA ligase can repair the single-stranded DNA molecules, or not double-stranded, only single-stranded DNA molecules repair. And the TOC DNA ligase will catalyze here also any type of ligase catalyze phosphodiester bond between two adjacent oligonucleotide which are hybridized to complementary DNA strand. Here the oligonucleotides mean they are the short single-stranded nucleotide consisting part, that is called oligonucleotides. Already told you, while the formation of new strand on this red strand, this is blue color new strand. In the new strand formation, here the maybe some portions of short strand can be missed. So this portion called as oligonucleotides. And this enzyme for this activity, for the ligase activity, requires NAD plus as a cofactor, like a E. coli ligase, E. coli DNA ligase. E. coli DNA ligase require NAD plus as a cofactor, and TOC DNA ligase utilizes only NAD plus as a cofactor, right? That means these are almost similar. But Taq DNA ligase only utilized for the synthesizing single-stranded DNA. Next, T4 RNA ligase. It is not an important one, but remember it. T4 ligase enzyme catalyzes the formation same phosphodiester bond between RNA and RNA oligonucleotides. But in Taq DNA ligase, DNA oligonucleotide. T4 RNA ligase, this phosphodiester bond formation between RNA and RNA oligonucleotide. RNA, DNA, RNA oligonucleotide or single-stranded DNA nucleotide also, right? And it requires ATP as a cofactor. But what about in DNA ligase? NAD is the cofactor. But T4 RNA ligase, ATP is a cofactor. And another different, someone trying to ask question.
T4 RNA మెగ్నీషియం కూడా అన్నారు కదా అని.
Magnesium common. Okay, but here changes in NAD cofactor, ATP cofactor. Hello?
Yeah, okay, sir.
Right. And it does not require template stand. But in Taq DNA ligase, it require template stand. Because already one template stand there, on the template stand newly synthesized stand, single stranded present. If the template stand present on the template stand complementary sequence, this same sequence can repair by DNA ligase. But in T4 RNA, without complementary stand can repair. So T4 RNA ligase can be used for variety of purpose, including construction RNA DNA hybrid molecule. How the formation hybrid molecule? DNA hybrid. Hybrid means nothing but some sort of recombinant combination. To different combination forms the hybrid. It's clear? Hello?
Yes, sir.
Yes, sir.
Right. So the ligases, mainly this ligase also the short answer questions. Ligases in short answer, you will have to write T4 DNA ligases, Taq DNA ligases, and T4 DNA ligases. 4 types enough. With comparison. To write with comparison. Okay? Clear?
Okay, sir. Okay, sir.
And kinases and phosphatases. The kinases and phosphatases I will explain in next unit because they are again repeated. That's why. And where it is after vectors. Mainly here. Genomic cDNA library, polymerase chain reaction, they are again utilized. That's why I will explain there. Okay. Kinases and phosphatases. Next one. Polymerases. Another important one is polymerases enzyme. This is also very important. Polymerases enzyme. The polymerases enzyme important not only in theory as well as in competitive purpose also. Right? Here the different type of polymerases enzymes are given. Polymerases. What is the function of polymerases? Synthesize newly stand. Just wait. Right, DNA polymerases are the enzymes that are synthesized new strand of DNA complementary to an extending DNA or RNA template. Already, which was explained in DNA synthesis and translation process. DNA polymerases can function only if the template passes a double stranded region that act as a primer for initiation of polymerization. There are three types of DNA polymerases are used in. They are DNA polymerase 1, clinofragment, and Taq DNA polymerase. Right. DNA polymerase 1, clinofragment, T4 DNA polymerase, or Taq DNA polymerase. Right. T4 DNA polymerase and Taq DNA polymerase. Here another also given T7 and MULV reverse transcriptase. So there are different type of polymerases are given. But just understand about the box in page number 118. Page number 118. Box. Page number 118 clearly given. One side DNA polymerases types: E. coli DNA polymerase 1, E. coli DNA polymerase 1 clinofragment, and T4 polymerases, T7 DNA polymerase, Taq DNA polymerase, MULV reverse transcriptase, different type of. And another hand, another column given the activity 5 and 3 and exonuclease activity, 3 and 5 and exonuclease activity, error rate, strand displacement, heat interaction, inactivation. So in this characteristics, most important one, first and second, 5 and 3 and 3 and 5 and. Right? Let's see one by one. First, let's discuss about DNA polymerase 1. DNA polymerase 1. This enzyme attaches to short single stranded region in a mainly double stranded DNA molecules and then synthesize a completely new strand, degrading the existing strand as it processed. This exhibits dual activity, DNA polymerization and DNA degradation. What is degradation? Polymerization means synthesizing new DNA strand, right? What is degradation? That means one end degrade the DNA and another end synthesize the DNA. Separated degradation means separated phosphate bonds, separated hydrogen bonds. Hydrogen bonds between where the hydrogen bonds? Hello? Hello? Hello?
Sir,
Where the hydrogen bonds? This DNA polymerase activities, the major template stand. What are the major templates for their activity? Polymerase synthesizes. What synthesizes? Synthesizes either DNA or RNA.
DNA.
Right? It can synthesize DNA, it can synthesize RNA also. And especially DNA polymerase 1 function, cleavage the hydrogen bonds. Hydrogen bonds means in this screen. So this is 5 and this is 3 and, right? Two start between water present, like a ladder like bonds. This one, this one, this one.
Yes, sir.
This one.
for diester.
So these are actual hydrogen bonds between
Nucleotides.
double bond, thymine, guanine, hydrogen bond, cytosine. So the cleavage of hydrogen bonds called as degradation. What happens if the degraded DNA? What happens if the DNA degrades?
No synthesis.
Synthesis of DNA.
That means separated DNA.
Hmm.
Separated DNA. So this activity, this degrade activity can direct 5N to 3N direction. See the box. That means 5N to 3N direction. For example, this is 5N to 3N. 5N to 3N direction or 3N to 5N direction. Both directions. And apart from that, it synthesize newly stand. It synthesize newly stand. So that is the important of DNA polymerase 1. And next one, the clue new fragment. It is also DNA polymerase 1 without exonuclease activity. It synthesizes a complementary DNA strand and single stranded template. The major application of the Clino fragment and related to polymerases are in DNA sequencing. Are you follow me? The second type of DNA polymerase does not involve in exonuclease activity. Not involved in exonuclease activity, but it can synthesize. What is exonuclease activity? Exonuclease activity, nothing but degradation.
Degradation of hydrogen bonds.
Right, right.
Okay, sir.
But here, DNA synthesis, it can synthesize newly DNA, but it cannot shows exonuclease activity like DNA polymerase 1.
Yes, sir. Yes, sir.
Here DNA polymerase 1 shows the activity, exonuclease activity, both direction, 5N to 3N direction, 3N to 5N direction. Right?
Okay, sir.
But few DNA Clino fragments sometimes show the exonuclease activity only 3N to 5N, but not both direction. Only 3N to 5N only. Right? But it is very rare. Okay?
Okay, sir.
Next. And what is next? TARC DNA. TARC DNA polymerase. It is a TARC means here the thermostable DNA polymerase isolated from the thermophilic bacteria, Thermus aquaticus. It will retain activity even after exposure to temperature. Actually, when this DNA denatured, when it is exposed to heat, for TARC DNA polymerase, which is extracted from Thermus aquaticus, even if we expose it to up to 90 degrees centigrade, it cannot denature. And it is central to polymerase chain reaction, one of the modern molecular biology's most important technique. That's why it is used in, because if you give any temperature, automatically denature means disactive, inactive, the polymerases. If polymerase is inactive, it can their activity, it can show their mechanism, it cannot. That's why where the high temperature utilization, you should use TARC DNA polymerase. A wide variety of polymerases have been categorized and commercially available. All DNA polymerases, almost all DNA polymerases mechanism added nucleotide to the 3-hydroxy group. Right? Always added to 3-hydroxy group. Added to 3-hydroxy group. Like it is shows continuation growth. This is called polymerization. Right? In TARC DNA, what is TARC? Right, TARC DNA polymerases also shows exonuclease activity. Exonuclease, where it is? As TARC DNA polymerase shows exonuclease activity. Right? But it cannot synthesize newly strand DNA. Is it clear? Hello?
So the point లేదు కదా ఇక్కడ.
Where?
Tap DNA.
Tag DNA?
అంటే ఎక్సోన్యూక్లియేట్ యాక్టివిటీ ఉంటుందని లేదు పాయింట్ అని.
Already told you earlier, just to see the box page number 118. 118.
ఓకే సార్, ఓకే.
Column number 1, 2, 3, 4, 5, 6th column. Tag DNA polymerase.
ఎస్ సార్, ఎస్ సార్.
Tag DNA polymerase. Are you able to see?
Yes, sir.
ఎస్ సార్.
And see the left column, the character 5 and 3 and exonuclease activity. Next 3 and 5 exonuclease activity. Next error rate, start replacement, and heat inactivation.
Yes, sir.
Okay.
Yes, sir.
Here the star mark. Star
Yes.
mark means present. Tag DNA polymerase 5 and 3 and exonuclease activity right. And 3 and 5 and exonuclease activity present or absent?
Absent.
Absent, sir.
Present or absent? Is there given star or not?
No,
Yes,
sir.
sir.
Absent.
No, sir.
What about E. coli DNA polymerase 1? E. coli DNA polymerase 1, 5 and 3 and exonuclease activity done or not?
Present.
It is there.
What about 3 and 5 and exonuclease activity?
It is there, sir.
E. coli polymerase clinofragment 5 and 3 and exonuclease activity?
No.
Only 3 to 5.
Only 3 to 5, but it is very rare.
Rare.
Right?
Yes, sir.
What about T4 DNA polymerase?
Only
T4
3 to
DNA
5.
polymerase?
Only 3 to 5.
T7
3 to
DNA
5.
polymerase?
Only
3
3
to
to 5.
Tag DNA polymerase?
5 to 3.
No, sir.
Reverse transcriptase?
Reverse transcriptase.
Reverse transcriptase, MMULV reverse transcriptase.
It is not.
Nothing.
Except heat
Heat
inactive.
inactivation.
Right. No exonuclease activities.
Yes, sir.
What about heat inactivation except Tag DNA polymerase? All are heat
Stable.
inactivation.
Yes, sir. Yes, sir.
Why Tag DNA polymerase resistant to heat?
Thermostable.
Because it cannot show any activity.
That's why I'm asking why.
And 90-degree temperature low, if it is exposed.
That's what I'm asking why.
Yeah.
Why it is resistant to up to 90 degrees centigrade?
I don't know.
Where it is extracted?
Thermophilic.
From the bacteria.
What is thermophilic?
The temperature stay.
Can resistant to temperature
At 90
because
degrees.
of it consists of the special DNA, Tag DNA polymerase enzyme.
Okay.
That is the nature of this enzyme activity. Right?
Okay.
Okay.
Clear?
Yes, sir.
Any doubt in this? Last, reverse transcriptase. In nature, this enzyme is involved in the replication of some RNA viruses.
Yes, Steve.
Retroviruses. This enzyme synthesizes DNA strand using the RNA.
No.
What is transcription?
Will
What is transcription? Already completed.
tRNA transcript.
Formation of mRNA.
Rewrite or change the.
Okay, formation of mRNA from
DNA.
DNA.
DNA. But reverse transcriptase, reverse.
Means RNA నుంచి DNA.
That's it.
mRNA.
From RNA to DNA form.
DNA. Okay.
That is reverse transcriptase. That's why it does not show any exonuclease activities.
Okay.
Exonuclease activities, right? Because this reverse to the DNA synthesis. This ability to synthesize a DNA strand complementary to an RNA template. Right?
Yes, sir.
Template.
Gene cloning vectors.
Characteristics.
Already applications of polymerases repeated always, repeated 5 into 3 and exonuclease 3 into 5 and exonuclease activities and polymerase activities. Polymerase activities means synthesize. Remember only two things you should remember. 5 into 3 and or 3 into 5 and any 5 into 3 always polymerase activity 5 into 3 and
Okay.
polymerase activity of
Exoneration.
always p and because already told you many times newly synthesized DNA, newly synthesized RNA, any one direction of synthesis always 5 into 3 and only.
Okay.
So 5 into 3 and polymerase activity means newly synthesizing DNA.
DNA.
And exonuclease activities means removal of hydrogen bonds. That's it. And also given some diagrams already explained in restriction endonuclease, how the blunt end formed, how the cohesive ends are formed. They are all already explained. Right? And next. Reverse transcriptase enzyme also and terminal terminations enzymes. These are all I will explain in PCR, formula chain reaction. RT-PCR. RT-PCR and PCR and RT-PCR. So in this that class I will explain later because here given only very short. RT-PCR, this RT-PCR long question. And also it is conceptual wise very interesting.
Reaction.
In the present COVID situation, you should know about RT-PCR mechanism and how it is work.
Okay.
Okay. I will explain later in PCR topic.
Subject.
Right, in terminal transferase also there and also nucleases also repeated there, but somehow again I will explain here somewhat about nucleases, but nucleases matter given here somewhat different, but I will explain in very easy process. Nucleases, the enzyme that degrades DNA. Nucleases, where are the nucleases enzyme first? Nucleases enzymes in nucleus which degrades DNA and RNA. DNA and RNA, that means it breaks the DNA strand, it can break the RNA strand.
RNA strand.
Right? There are different type of DNA, different type of nucleases. That is DNase, RNase, DNase 1, ribonuclease A or RNase A. They are different type. So these mainly the most using nucleases DNase 1 or deoxyribonuclease 1 and another one is ribonuclease A. These both are purified from bovine pancreas, that is cattle pancreas. DNase 1 cleaves double stranded and single stranded DNA. But remember one thing, it is cleaved between phosphodiester bond. Let me show the diagram.
Nico.
Are you able to see this?
Was so small.
Yes, sir.
Small.
So this is sugar, right? This is one phosphate, right? This is another phosphate, right? Okay. Sugar, phosphodiester, two ester bonds. And here also same sugar, sugar 1, sugar 1. Here only hydrox group. But let's see here, sugar S, phosphodiester bond. Diester bond between one sugar molecule to another sugar molecule. So this bond removed. As a result here, separated sugar. So actually this is the without separation. Without separation. When we use DNAase or RNAase, it is separated into two phosphodiesters like this. Like this. How many phosphodiester bonds here? 1, 2. Here 1, 2. Like so, the cleave are separated phosphodiesters bonds. Okay, is it clear?
Yes.
Right. Now the next gene cloning vector. Are you any disturbance to you?
No, sir.
Okay. The outside some drums are any drum sound?
Yeah.
No, sir.
Okay, thank you. Next one. Gene cloning vectors. Vectors also very, very important in theory and competitive level. Vectors where methylase activity and RICA protein, aggress enzymes. I will explain later because they are actually ఈ ప్లేస్ లో ఉండే కావివి దాని టాపిక్ రిలేటెడ్ వచ్చిన దగ్గర అక్కడ ఎక్స్ప్లెయిన్ చేస్తా ఓకే హలో
Okay, sir.
రైట్ క్లోనింగ్ వెక్టార్స్ I told you gene transgenic plants. What are the vector? What is the importance of vector? What is the cloning vector? What is the important? What are the characters of the vectors? After that, let's discuss about types. Vectors small, independently replicating. Why we use vectors? Because actually you separated a desired gene. You want to get that type of desired gene more number. If you insert into vector, that vector should consist the character of replicating, self-replicating, so that the desired gene also replicate many time. Right? Hello?
Yes, sir.
Yes, sir.
Right. So it is a vector is the small independently replicating genetic element that is called cloning vector. It used to replicate the genes and also used in cloning. That's why it is called as cloning vectors. Cloning vectors are generally designed to allow recombination of foreign DNA at restriction site. Already told you how the foreign DNA inserted into the vector. Okay. Hello.
Sir.
Right. Some of the characters should be consist to the vector, such as vector must possess an origin of replication. That means replication site should consist replication site. If you selected origin, what if there is no replication origin? You selected the vector, but no origin of replication. Without origin of replication, can it replicate?
No, sir.
Right. That's why it should consist origin of replication. The vector should replicate several times per cell cycle, giving rise to the high copy number of vector per cell. The another character, it can already given the definition, independently replicate. Independent replicate means in per second, it gives number of copies. Number of copies means here number of copies what? Which copies?
DNA.
Which DNA?
Pick.
Which DNA inserted into vector?
Desired.
Desired genes. It should possess restriction site for range of restrictions enzymes. Restriction site. What if there is no restriction site?
No
No
replica.
restriction. Can you cut the vector gene?
No.
It will
Without cut, can you join the desired gene?
No,
No.
sir.
That's why it should possess restriction site. Restriction site convertible to cut some portion of the vector gene. At the same place, insert the desired gene.
Yes.
And the vector should be comparatively small in size. The
Okay.
vector must be easily introduced into host cell.
Okay.
The vector should have one or more selectable markers.
Gene markers.
Yes, gene markers I will explain later. So these are the characters. And what are the vectors in general?
Bacteria.
Cloning vectors.
Bacterium.
What is the, okay, bacteria also vector, but which is extracted from bacteria, which is self-replicated gene in bacteria. Self-replicated gene in bacteria. So the different type of vectors: plasmids, bacteriophages, cosmids, and fog plasmids. Plasmid vector. Tell me. Hello? Just wait. Are you able to see the diagram?
Yes.
Plasmids.
Yes. Bacterial plasmids are small circular, which is present in bacteria. If you know the bacteria structure. This bacteria cell, right? bacterial cell, which is plasmid, which is bacterial genome. So this is the plasmid, you have to extract it. So bacterial plasmid are small circular DNA molecules that are not only distinct from main bacterial chromosome, but also additional to it. And when we compare with bacterial genome, distinct from bacterial genome, and it is the additional part, extra part, that is simply called as extra genome in bacterial cell. It can replicate independently. Many different types of plasmids have been found in bacteria. The plasmids that are routinely used as vectors and those that carry genes for drug resistant genes useful because drug resistant phenotype. Because actually, I will explain later the property of this plasmid. But here the several different type of bacteria extract plasmids extract from bacteria. Among that, the most important one is PBr 322. PBr 322. Plasmid PBr 322 is simpler in structure. It has two drug resistance. They are TET RER. TET or here we can see. That is tetracycline resistance. And AMPR, that is amplicillin resistance. What is amplicin tetracycline? Bactericides. They are bactericides. But this, if the PBR plasmid present, if you use the amplicin tetracycline, does not harm to the bacteria because it has the resistance gene. Okay. Right? The TETR and AMPR, both genes contain unique restriction target site that are useful in cloning. Right? That means, for example, already the bacteria resistant to the amplicillin tetracycline. Right? So some organism, some extra any other organism, the frequently infected with the infected bacteria which are produces amplicin and tetracycline. Amplicin tetracyclines causes the harms, damage the organism. In this situation, why it is harm the organism? Some bacteria causes diseases other organisms. Why can't damage the same bacteria? Hello? Hello? Hello?
Sir.
Are you follow me?
Yes, sir.
My question is, here one organism, for example, human being, some tetracycline bacteria, that means tetracycline production bacteria or amplicillin production bacteria, if invade the human being, it cause diseases because which releases amplicin, tetracycline, which both chemicals harmful to the human beings and human cell. It can resist to the amplicin tetracycline. Human cell can resist for this to those chemicals, cannot resistant because this resistance power only have the PBr 322. This PBr 322 plasma is present in animal cell. Hello? Is it present in animal cell? Hello?
No, sir.
Right. Therefore, the cloning produces is to mix with samples, cut the plasmid and donor DNA. That means, for example, do you want to select this region, amphicycline region or tetracycline region, these two. Just cut these two plasmid genes, separate it, okay, and join with another DNA gene. Cut and join with another DNA gene. Later, what happened after joined? Recombinant DNA formed. Hello?
Sir.
Recombinant DNA formed. And this recombinant DNA, after formation, recombinant DNA, further experiment are needed for find the clones with specific insert required. That means after formation recombinant DNA, this recombinant DNA inserted into again. దేంట్లోకి ఇన్సర్ట్ చేయాలి ఆర్డిఎన్ఏ టెక్నాలజీ స్టెప్స్ నేర్చుకున్నారు కదా
yes sir
ఏం నేర్చుకున్నారు
వెక్టర్ లైన్
ఫస్ట్ డిజైర్డ్ జీన్ సెపరేటెడ్ వెక్టర్ జీన్ సెపరేటెడ్ రైట్ హలో
yes sir
first in RDNA steps you have to select desired gene, separate it. Vector plasmid, separate it. Both are joined as result formation RDNA. Now RDNA, after formation RDNA inserted into
host, sir.
What is the host here? Okay, where the plasmid replicate, self-replicate, in which cell? Hello?
Self-replicate in bacterial cells.
Bacteria. That's why again after recombinant DNA formed, again inserted into the bacteria. What happened after you inserted into bacteria? This RDNA, actually you inserted only one RDNA, recombinant DNA. This one recombinant DNA, apart from the, along with the bacteria cell, it becomes, already told you what is the property of plasmid?
Self-replicating.
Self-replicating means single copy becomes
multiple copies.
Multiple copies. To get multiple copies, again this RDNA inserted into another host bacteria cell. Okay? Hello?
Yes, sir.
Right. Next, bacteria phases. What is bacteria phase? In here given clearly page number 131, 132. In 132, figure number 9.3. What happened? Figure number 9.3. One bacteria becomes. Hello, see the diagram. Are you follow the diagrams?
Yes, sir.
What you follow? Hello? Page number 132, figure number 9.3. One bacteria cell becomes two, but not given here. These two individually, if it is replicate, how many cells are formed? Again, that means each one again two, total.
Total 4.
If this 4 replicate, how many?
8.
That's why it's multiple number, continuous multiple, right? Okay.
Yes, sir.
Right, and let's see the other different types of vectors. F plasmid, resistant plasmid, already resistant plasmid completed. coal plasmid, degradative plasmid, virulence plasmid. F plasmid, F for fertility. Fertility means which is useful in gene transfer from one through conjugation mechanism from one bacteria cell to the another bacteria. What happened here? Just wait. Bacteria, conjugation tube, bacteria conjugation tube. So this is F plasmid, this is bacterial chromosome. So this F plasmid self-replicate and convert into another bacteria. Here already bacterial chromosome present, but not F plasmid. When it is migrated into this one, here also self-replicate again formed into plasmid. Right? This type of plasmid called as F plasmid. Resistant plasmid already completed, that is amplicillin resistant, tetracycline resistance. Coli plasmid, which contains the genes that code for determine the produce of bacteriocin, proteins that kills the bacteria. Coli plasmid, there's some production of taxins. The function of coli plasmid, production of taxins. This taxin kills the other bacteria. Degradative, name given, degradative plasmid is unable to enable the digestion of unusual substances. Virulence plasmid also cause the diseases. So which turns the bacterium into the pathogen, that is cause disease. Virulence means virulent property causing diseases in humans and plants, like that. Right? Now the bacteriophages. Bacteriophage means viral vector in which the genes or gene of interest are incorporated into the genome of virus. Bacteriophage, viral and bacterial, both genes are present.
Yes.
That means artificially they are formed. But first of all, what is bacteriophage? Bacteriophage is the virus. Remember it.
This which
Bacteriophage is the
infects-
virus which infect the bacteria. That's it. Bacteriophage is the virus that infect the bacteria. This virus consists the genome, that is bacteriophage. This bacteriophage also utilized as a vector. The most widely used bacteriophage M13 based vector. Phase lambda vector. Let's see here in Cosmic P7. Page number 136. Page number 136. Bacteriophages vector. Bacteriophage M13 bacteriophage vector, two different type. Lambda phase vector. M13 is a filamentous bacteriophage of Escherichia coli and contain, no need this KB, no need this structure. Just wait. Clear? Are you able to see? So this is bacterial phase vector. Sir, where is M30? Okay. Lambda phase. M13 bacterial phase. Where is the diagram? Okay, right. Oh, shit. Okay, not downloaded. Okay, sir. No, bro, I will explain later. I will explain next class. What about other classes? Hello? 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 Are you in line? Hello? 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 Am I audible to you? Hello? 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 Hello?
సార్
వినిపిస్తుందండి?
వినిపిస్తుంది సార్. మాకు స్క్రీన్ ప్రెజెంట్ చేయకపోతే రావట్లేదు సార్. ఈ స్క్రీన్
స్క్రీన్ కాదు, నేను వాయిస్ వినిపిస్తుందని అడుగుతున్నా.
వాయిస్ వినిపిస్తుంది మాకు మ్యూట్ చేయడం రాలేదు సార్. మీ
నేనేం
ప్రెజెంటేషన్
మ్యూట్ లో పెట్టలేదుగా మిమ్మల్ని.
కానీ నాకు రాలేదు సార్ ఆప్షన్.
అన్ని క్లాసెస్ మంచి నడుస్తున్నాయా?
ఎస్ సార్, అన్ని నడుస్తున్నాయి సార్.
మరి ఎందుకు జాయిన్ కావట్లేదు? ఎవరు జాయిన్ కావట్లేదు ఎందుకు?
5 members వస్తున్నారు సార్ ఒక్కొక్క క్లాస్ కి, పెట్టినా వెంటనే ఎవరు రెస్పాండ్ కావట్లేదు సార్.
మీరు 5 members
వాట్సాప్
లేరు కదా, చాలా మంది ఉన్నారు. మీరే కదా రిక్వెస్ట్ చేసి మరి నైట్ క్లాసెస్ కావాలన్నది, ఈవినింగ్ క్లాసెస్ కావాలని చెప్పింది మీరే కదా.
అవును సార్, నేను అంటే 5 to 6 members జాయిన్ అవుతున్నారు సార్. వాట్సాప్ లో
మీ దాంట్లో
మెసేజెస్
చాలా మంది ఉన్న వాళ్ళు మీ టీచర్స్ మాకు డే టైమ్ లో కుదరదు, ఈవినింగ్ టైం అయితేనే పర్ఫెక్ట్ అన్నారు. ఇక్కడ ఎవరు రావట్లేరు కదా మరి దీంట్లో.
డైలీ 6 members 7 members 5 to 6 members వస్తున్నారు సార్.
5 to 6 members కి అయితే మేము యూనివర్సిటీ టైమింగ్స్ లోనే పెట్టేవాళ్ళం, సాటర్డే సండేస్ కంటిన్యూషన్.
సార్
ఇట్లా పెట్టి యూజ్ లేదు కదా, రిస్క్ తీసుకుని పెట్టడం ఎందుకు? సరే ఓకే అండి, బాయ్.
ఓకే సార్.